Hello, today we are going to construct the phylogenetic tree by using MEGA tool. MEGA that stands for Molecular Evolutionary Genetic Analysis. MEGA that is an integrated tool for conducting automatic and manual sequence alignment, inferring phylogenetic trees, mining web-based databases, estimating rates of molecular evolution, and testing evolutionary hypotheses. So today we will learn how we can construct the phylogenetic tree by using MEGA tools. To construct the phylogenetic tree, so here four basic steps. The first step is that identified and acquire a set of homolog sequences. We should have the sequences which we can use to construct the homologs phylogenetic trees. Second, we have to align the sequences. And the third, we have to estimate, either we can say that the construction of phylogenetic tree. When we have the phylogenetic tree, then how we can present to clearly state what we are going to extract the information from that phylogenetic tree, and then we can inference from that tree. So these are the four basic steps to construct the phylogenetic tree. The first step, to retrieve the sequences, we should already have the sequences which we will use in MEGA. The second step, align the sequences. It can be used by any other specialized alignment sequencing methods. Either you can also use the MEGA tool. The third, construction of phylogenetic tree, and we can also use MEGA for the construction, and then we will have a phylogenetic tree. And then for customizations, either you can go to any other tools, or you can also use the MEGA. So first of all, the first step is that we have to download and we have to install the latest versions of MEGA Molecular Evolutionary Genetic Analysis tool from its website. So we can go to that website. So that is megasoftware.net. So here you can see that from here you can download according to your operating system, either you are using Windows, Mac, Ubuntu, or any other type of the Linux system. So graphical units command, and either you are using the 64-bit or 632-bit Windows, and then you can download. After the download, you can install the Mega Tools. So after the installations, you will have the you can open. So this is the interface of the Mega Tools. So here you can see that these are the, either you can access from here, you can go from here, analysis, help, file, either these are the shortcuts for the Mega Tools. So what's our main purpose to construct the phylogenetic tree? So the second step, first step we already completed, that we have the sequences. Okay, the second step, now we have to construct the alignment. So now we can go from here, edit or build alignment. From build alignments, so here is the option, either you want to create the new alignment, either if you already have the alignment, then you can open a saved alignment session. So here we want to create a new alignments. Here, so it will ask you either you are building a DNA or protein sequences. So we will choose, we have already retrieved the protein sequences which we will use to alignments. So here it will show the protein sequence alignments. So we can, so we already prepared the sequences of these genes, ING1, homologs, orthologs from chimpanzee, same gene from elephant and other some species. So we can press Ctrl A to select all of the species. We copy it, and then here in this alignment session interface, so we can just paste the sequences. So here we can see the ING1 human, chimpanzee, from elephant, from rat, from horse, zebrafish. This is just like, so here we can see these are the sequences we already have. So first of all, here you can go to the alignment section, alignment by cluster W by muscle. So we can choose cluster W method. So it will select and then it will ask you about the parameters. So it's up to you which parameter you want to modify, or you can only go with the default parameters. So we can just click. So it depends upon the number of the sequences. If you have too many proteins, so then it may take more time in alignments. So now we have done with the alignments, so we can save the alignment, right? So now we can quit this alignment explorer, right? So here this is the main. So either you can go from here to construct the phylogenetic tree, either you can also see the shortcut here. So these are the phylogenetic tree construction methods. Either you can use maximum likelihood, neighbor joining, minimum evolution. So we can select the first one. So it will ask you about the saved alignment. It will be used. So here we have. So now it will ask about the analysis preferences. So here bootstrap method. So we can, like, just to verify the phylogenetic tree. So it's up to you. Either you can use without bootstrap method, either you can use the bootstrap methods. Okay, so here, so without bootstrap values, and either you can use the bootstrap for, like, maybe it will take time. So we can use 100. So then you can, either you want to subset to use, either you want to complete deletion, use all sides, and uniform rates for rates, the inference option. So it's up to you, you can choose. So here, so because we have selected only 100 bootstrap values, if you select, like, so mostly it is recommended to use 500 or 1000 bootstrap values to construct the value genetic, to confirm, to validate either they have the same replication or no, right? So now we have constructed the phylogenetic tree. So here you can see the further parameters to customize these phylogenetic tree. So here you can see, if you want to increase the font size, if you see that if you have many species, like, you cannot see the names or labels of the species, you can increase the font size, and even you can also change the font style. So then, like, from here, if you want to change the layout, tree style, for example, this is the traditional. If we want to straight curved radiations, either you want to change into circle. So here you want to change, and then auto size tree, if you want to adjust into a default root on midpoint, if you want to, like, tree width, if you want to decrease or width their height, you can do this one. And then you want to subtree, if you have a subtree, from that if you want to add something. And then statistics, frequency informations, distance, scale, okay, ancestors, computes, like, according to the nodes. So these are the many things, if you want to zoom in, zoom out, or you want. So when you have constructed the phylogenetic tree, right, so then you can save to clipboard. Either you directly want to save into the Microsoft Word, or any or PPT you are using, either you can save as a separate file in PDF, PNG, score vector, TIFF, or EMF. So you can select which one having the good resolution, either like a TIFF, EMF, and SVG. Even you can also use the PDF. So here you can construct the phylogenetic tree, and then you can save in a separate files. So there is another thing, if you want to export current tree in Newegg, just like a, the any other phylogenetic constructions methods, either the customizations tools, so you can export in to this one. So here this is like, the new export. So here you can copy this one,